Uh, Hello. I'd like to thank you guys for letting me speak today. This is an amazing symposium, and I really look forward to talking to you all individually as well. Uh, I am working with Arthur Olson in the lab at Scripps Research Institute and on the CellPack project. And today I'm going to talk about a specific application of CellPack that we're developing, which I think is going to be very useful. And that's the making of synthetic cryo-electron tomograms. So you all know about cryo-electron tomography. We've talked about it a lot today. It's a very hot field and developing quickly. One of the things that needs to be developed are computational image analysis methods. CellPack, of course, builds structural models from PDB files. And we think that these two are natural to go together. You've seen what CellPack can do from the large scale down to the atomic level modeling. Remember this picture for later because I didn't put it in. So one of the things that CellPack is really good at is integrating data sets. So for this specific project, we're using tomograms, proteomic studies, molecular structures from the PDB, and literature review. But I think you can all see how CellPack could also be used to integrate SACS data and pretty much any kind of data that you can throw at it. And so that's one of the very exciting things. And you put all that data together in what's called a recipe, and then that recipe is it establishes the parameters for the cells. And then from that recipe, you can make as many models as you want that are all variations of those parameters. Now, up in the left here, we have a cryo-electron tomogram. And one of the things that is a real challenge in tomography is the segmentation and annotation of the tomograms. And that's usually done by hand, but there are numerous people working on automated methods of doing that. The bottleneck here is that we can't know for sure what we are seeing, and that is that is what we're trying to address. The general process for finding the complexes in these tomograms is template matching, or there are also template-free methods that are being developed by Albert's lab right now. And the problem is, if you take a template or a template-free method on an actual tomogram, how do you know if you're correct, if you got it correct? The ribosomes, perhaps, ATP synthase, you can know fairly accurately, but for other molecules, it's a very rapidly developing field. And so what we want to do is create synthetic tomograms from CellPack, where we can fill in all the blanks with the proteins that we know should be there, and then create synthetic tomograms there where all of the proteins and structures are completely known. So we have a ground truth that we can be based off of, and then we can use this synthetic tomogram to train algorithms and also to test new methods of segmenting and annotating things automatically. So Frank Albert's lab is at the forefront of this, and his program, TomoSimu, is what I based my work on. I have integrated TomoSimu and CellPack so that now we can create a synthetic tomograms at least the same level. And you can see that they're at least very close. This is at the very early stages of this development. This was just made last week. But we are hoping that we're going to be able to take it further. And for example, this is a cell, and we're a pilot project. We're working with the Craig Benter Institute. Their synthetic organism, which is based on Mycoplasma mycoides. It's a minimal genome organism. So we're trying to start with the simplest possible cell that we can go with. And it's basically just a membrane with a bunch of proteins and some DNA in it. We have done the experimental image, segmented it, which is basically just putting a membrane on it at this point, filled it in with CellPack, and then made a synthetic CET off of that. And as you can see, there are some similarities, at least, between the two. You can spot the ribosomes that would be identified by hand in the experimental and also that are there in synthetic. So the things we need to do are improve the quality, improve the speed, integrate more fibers, DNA. These are all big challenges, but also implementing automation so that we can gather all of the information that we want to get, as Art was talking about. And that's one of the things that I'm really looking forward to talking to all of you about, because I think that we can do it, and I think that we're on the way. So things are going to get better and better. And this is a thank you to all of the people that have been involved in this project.